Hi everyone, I know it's a long time, but let's start chapter 5 Principles of Inheritance and Variation from class 12. So, welcome back to Biology 101, and this is Amrita. So, we all have wondered in our childhood, right, how a baby elephant comes from elephant, not a lion or anything else, and a mango plant from mango seed, and offsprings are always identical to their parents, right? Now, why so? This is due to the inheritance. What is inheritance? Process by which characters are passed from the parent to progeny. Progeny are the kids or the children, right? Now, basis of inheritance is the basis of heredity. This is the process by which characters are passed. Like you can get some characters from your mom and some from your dad. You are not exactly the same. That is what we come to variation. Variation means there's a degree by which progeny differ from their parents also. It's not like you are the exact copy of your parents, right? You inherit some characters from your mom and some characters from your dad. Now, humans knew cause of variation is hidden in sexual reproduction. That is back then 8000 to 1000 BC itself, humans came to know that this cause of variation is hidden in the sexual reproduction itself. Now what did the humans do? They exploited the variations in wild populations of plants and animals. They tried to investigate and to find out how this is happening. So they exploited the variations in plants and animals. Of course, they cannot do in the humans. So they selectively bred to get desirable characters. How do they do it? So let's say we have a cow A and cow B. Cow A has good yield in milk. Now cow B has good meat. So what do? They take gene from this of the good yield of milk and gene from this of the good meat, and they cross it. They cross the both of the cows to get another cow which has both of these characters. So that is where we get the desirable characters. Now for example, through artificial selection and domestication from ancestral wild cows, Sahiwal cows in Punjabs are brought up. Now, we'll learn how each and every process is done in the upcoming chapters. That is not much important now in this chapter. So, let's go to the next topic, that is Mendel's laws of inheritance. This is the main part, and then the real laws are coming up in the next parts. Now, who is this? He is Gregor Mendel. He has given so much information for the genetics that we are learning now. He is the main milestone which he created. Now, hybridization experiments on garden peas. This is the plant he used, garden peas important. Now, 1856 to 1863, 7 years he had taken for this, and this year is also important. Now, what did he do? Propose laws of inheritance in living organisms, statistical analysis and mathematical logic applied to problems in biology. Before him, no one even tried to use mathematics and logic to solve the problems in biology. So he started that first. And these are the three main importance of what he did. Large sampling size, which gives high credibility to data he collected. There are large sampling size means so many of different variations, like so many examples he had taken, so many he had recorded things for his experiment. Sampling size gave general rules of inheritance, investigated two opposite traits. Example, opposite traits means if one is tall, opposite should be dwarf, that is smaller. Otherwise yellow green seeds. Now see, we have the characters and the dominant recessive trait. He had worked for 7 different things, right? Like seed shape, seed color, flower color, pod shape, pod color, flower position, stem height. All these 7 characters. Now in seed shape, round, dominant and wrinkled recessive. Seed color, yellow dominant, green recessive. Flower color, violet dominant and white recessive. Pod shape, full constricted. Pod color, green yellow. Flower position, axial, terminal. Stem height, tall, dwarf. Now we should remember this all. This is very important. Don't confuse between seed color and pod color. In seed color, yellow is dominant, green is recessive. In pod color, green is dominant, yellow is recessive. We can just remember it like whenever we see a pea plant, it is always in green in color, right? The outside part is green in color. So the dominant trait of pod color is green, because we have always seen that. We can remember it like that. Pod color is always green. that is dominant trait, and the recessive is yellow. But in seed color, it's yellow for dominant and recessive it is green. Now set a this opposite two traits he had investigated, right? That set a basic framework for other scientists to work on it later. He only told that we can compare both of the traits and do the experiments. So that gave the other scientists who later worked on this a basic framework. Now Mendel conducted artificial pollination or cross pollination experiments using true breeding pea lines. What is true breeding pea lines? Which has undergone continuous self pollination. That is, one trait is like a plant is always crossed with the same trait, like one plant has a trait and another plant has the same trait, so they are crossed again and again. That is self pollination. Undergone continuous self pollination, which has stable trait inheritance and expression for several generations. Now for the after the that self pollination, if they are again and again self pollinated for several years, they'll have the same traits, right? So Mendel selected 14 true breeding pea plant varieties, 7 actually, but 14 including the contrasting characters. That's what I have shown in this table. This is already given in textbook. Just a disclaimer, everything whatever I have written here is exactly in the NCERT textbook. I have read those lines and I have made notes. So you can make notes from this, or you can just listen to the lecture and go make your notes again. So coming to the next part, inheritance of one gene. Now what happens here, example, tall and dwarf are crossed and they are collected, the seeds of this plant are collected to learn about the first generation. We also call this F1, that is filial generation 1. Mendel found that all progenies were tall in the F1 generation. That is, Mendel made similar observations that F1 progenies always represent either one of the parents, that is either tall or dwarf, but here tall is the dominant one, so all the progenies were tall. Now how did he do that? Self pollination, that is he crossed now for the F2 self pollination of F1. Now F1 cross F1 will give F2. In F2 he found a difference from the F1, that is F2 he found some dwarf progenies, that is in F1 we had only tall, right? But in F2 we have tall as well as dwarf. Now this gives a conclusion that recessive characters show up in second generation but not in the first. That is 1/4 will be dwarf and 3/4 were tall. That is 3:1 dominant is recessive. Don't get confused, I'll tell in detail. Now tall and dwarf, all parental, there's nothing mixed, like nothing in the between. Tall, dwarf, they are the parents, right? Now both in the progenies also tall and dwarf only comes, nothing in the middle height between tall or dwarf. Now that's why there again no blending. In both F1 and F2 there is no blending, either it's tall or dwarf or both tall and dwarf. That is nothing in between height. Okay. So Mendel said something stably passed down, even he didn't know what, so he just gave that name as factors. He said something is stably being passed down from the parents to the children through the gametes, and that is factors. But now we know that is genes. They contain information of the trait that should be passed. These genes contain the information of what is to be passed. Now if your father has curly hair and you get the curly hair, the gametes will have the genes of the curly hair, the gene that will contain the info of which trait is going to be passed to you. Now that can be fair or height, skin color, height, or the curly hair, the texture of the hair, the eye color, whatever, everything comes from the parents. Now genes are the units of inheritance. That is, now here we already had learnt a line, right? Basis of heredity is inheritance, and now here genes are the units of inheritance. It's not very confusing, but just keep these two lines in mind separately. Genes are units of inheritance. Genes which code for contrasting traits are alleles. These alleles mean slightly different forms of the same gene. Alleles are slightly different forms of the same gene. So genes are units of inheritance. Now how do we represent the genes? How do we represent the alleles? T, we have written capital letter T and small letter t. Capital letter shows dominance, small letter shows recessiveness. Now for example, T and T are for tall and dwarf, for the height characteristic. Uh, that is the capital T for tall and small t for dwarf. These are alleles of each other. We have seen here, right? The slightly different forms of the same gene are called alleles. So here this capital T and small t are slightly different forms of each other. That is, they come under same height, but they come under the same characteristic height, but tall and dwarf. So these are alleles of each other. Now this can combine into three forms: capital T, small t, and capital T, small t. These are the three combinations which can come. So when it is both capital T, it is tall. When it is both small t, it's dwarf. When it is one capital, one small, it is also tall. It is only tall, it's not dwarf. So when it is both capital or both small, it is known as homozygous, and one capital, one small, it is heterozygous. These are the genotypes, and this is important. Don't confuse genotypes and phenotypes. Phenotype means physical appearance, genotype means genetical appearance. Now genotypes, we know this is the genes they have. So T, small t, big T, like that we just represent it. And phenotypes, we can see and say the phenotype, but not the genetical representation. So if a plant is tall, we can say the phenotype of this plant is tall. If it is a dwarf, the phenotype of this plant is dwarf. That is the genotype and phenotype. Cross between T that is dominant and recessive, that is the tall here and the dwarf here, is the monohybrid cross. That is monohybrid. What is mono means? One, we know. That is one character. Now next we have dihybrid. That comes when there are two characters. We will come to that next. Now I told, right, here we have 1/4 dwarf, 3/4 tall, 3:1 dominance is to recessive. Now we can understand how we did that cross and how we found it 1/4 is dwarf and 3/4 is tall and all. So in the textbook itself we have given, I have written side in the side just to give a clarity. Now this is how they cross it. Tall, we have T, that is capital T, dwarf, we have small t, they are crossed, that is selfing. In the F1 generation, we get tall and tall only. That is the F1, right? We said in F1 we have all the tall, only tall we get. Now in TTC, one T from here, capital T, one small t, one capital T, one small t. We got T plus TT, that is capital small. So both are tall, heterozygous, right? Now both are tall. Now let's see, this is F1 generation. Now if we self F1 generation, we get F2. That is, this is F2 generation. Now this is the F1 product, right? This is the F1. So if we self that, capital T, small t, one capital T, small t, small t. So we get, this is purely tall, this is heterozygous but tall, tall. Now we have two small t's, so that is dwarf. Now that is what I was telling. We have 3 tall, 1 dwarf out of 4. So 3:1, 3 tall, 1 dwarf. That is the, this is the genotypes as I was telling, and 3 tall and 1 dwarf, that is the phenotype. So this is, we got here the tall dwarf. This is the same thing they have shown here. Now this is monohybrid cross, is given in the textbook. They have also crossed like what I have done. Now a phenotypic ratio 3:1, genotypic ratio 1:2:1. How do we find this? Let me tell you. In this is in which generation? F2 generation. F2 generation only we can see the difference, right? So in F2 generation, let me tell you, this is tall, okay, tall and dwarf. Now what do we see? We see this is, when we come to phenotype, we can see 3 similarities, 1 non-similar. That is 3 similar, 1 non-similar. That is 3 tall and 1 dwarf. So this is the phenotype. That is physically we can see tall, dwarf. Now when it comes to genotype, see here, these are the genotypes. So we have one T, big T, and two similar, one big T and one small t, and one another different ones, both small t's. So 1 here, 2 here, 1 here. So 1:2:1. This is the phenotypical ratio. That's what they have given here. This is important. Many questions you can see even in your school or in exams, other exams. So next, this is called the Punnett square. That is British genetist. That is Reginald C. Punnett. Graphical representation to calculate the probability of all possible genotypes of offspring in a genetic cross. That is what this diagram representation and all. We need not draw all these boxes, but they have told us this is what we usually do if we get a question and all. So next, in F2 generation also there is no blending. As I already told, it's either tall or dwarf. There is nothing in between. So there is no blending. So by process of meiosis, alleles segregate. Now what is segregation? This random process, the segregation is a random process. That is 50% chance of gamete to get into either of the allele. T has both big T allele, small t has both small t allele. So it can go into the gametes by 50% chance. This symbol represents female, that is egg, we all know, and this symbol for male, and that is pollen. Now during fertilization, this big T from pollen, and if small t from egg comes, the zygote will have big T small t. That is heterozygous, but it is tall. That is how it comes. It has 50% to go into whichever gamete it wants, 50% chance, and T from, if it gets both big T from the pollen and from the egg, then it will become big T or small t. Does not matter. It's just the process. Now heterozygous, if this is the case, then it is heterozygous, big T small t, but the phenotype is tall. Pollen grains having big T has 50% chance to pollinate with small t in egg, and or to pollinate with capital T in in the egg itself. So here it is T. Okay, that's right now. So 50% chance to pollinate with small t in the egg, or 50% chance to pollinate with capital T in the egg. Okay, now next coming. By simply seeing a tall plant, we cannot be sure of the genotype. Right, now we have a plant in front of us. Now it is tall, we know, but we cannot say if it is capital T or capital T and small t, because both of them will seem to us as tall. By seeing the plant, we can never judge the genetic type, that is genotype. So what did Mendel do? He did something called test cross. This is very important. These diagrams will be given and they will be asked. Each and every line of NCERT is very important. Please try to learn it by the course. When you just go, don't think of it as a burden. Read it again and again. I know it will be hard, but you can do it. So Mendel did something called test cross. That is dominant phenotype into the recessive part. So dominant phenotype, of whose phenotype is to be found. We don't know this, right? We don't know what is the phenotype, genotype of the plant. So we are crossing that with a recessive parent. That is how we do test crosses. We cross the unknown one. If let's say X is the unknown one. Okay, let's say Y is the unknown one. So Y crossed with the recessive plant, the recessive one. So from this we can find what is Y. So let's say this is Y, right? So this is crossed with the recessive, and this is crossed with another recessive. To find, there can be two possibilities. If this is a double, they have made the color of the flower here. So if we say W for this violet one, and small w for this white one, that is recessive. So this Y can be capital W or small w and capital W, because both will be violet color only. So if this is capital W, when they cross with like this W into small. So if they do that, then we can see all the four will be dominant only. If both this is purely dominant, that is homozygous, and this is homozygous recessive, then all the progenies will be dominant only, that is all the 4 will be violet, all flowers are violet, which will give us a conclusion that unknown flower is homozygous dominant if this is the case. So we have first case here. Now second case, if the flower is W, capital W and small w, we have 2 violet and 2 this white. So how do we find that? When we cross W, I have already said, right, W cross small w. So this is what crossing takes place. So we get W. You can see the screen. So this is what we get. So 2 white and 2 violet. That is what they have given, 2 violet and 2 white. So half of the flowers are violet, half of the flowers are white. So unknown flower is heterozygous. That is all for today's video, and I'll keep posting continuously. We can see the next topics in the next video. Thank you for joining. So I think we missed on this topic, that is steps in making a cross in the bee. We talk about cross pollination or self pollination, right? So how do they exactly make a cross? So this we have flower here. Now first step will be removal of the anthers, that is this step is emasculation, first one. That is we do the emasculation, that is removal of anthers in the female one, and then in the male, from the male we transfer the pollen grains to the female flower, and that is how this is the pollination process, that is second one. Now due to the pollination we get the this product, that is both from the male and from the female we get the product together, that is this pea pod.